Hi guys, welcome back. Here's the last little mini lecture from chapter one. This little lesson is going to take a look at kind of the last few pages of chapter one, talking about what is science, how do we do good science, how was it done in the past. So just some thoughts on some of these ideas. So when we usually talk about science, a lot of times in beginning science classes, you're introduced with what's called the scientific method. You're going to be doing a lab this week with scientific method, gummy bears, soaking them in water and all that kind of stuff. But in that process, hopefully you're learning the idea of what a hypothesis is, how to set up a good experiment to maintain, control your variables and only focus on one idea at a time. And so to help scientists make sure that the results that they have are valid and can be used to explain phenomena is we follow what's called the scientific method. So the scientific method is usually the series of steps that you follow to get good, reliable results. So usually you start off with identifying a problem, making an observation. So then you are like, oh, the world, something, you make an observation. And then you form what's called a hypothesis. So a hypothesis is, there's lots of different definitions. I like to say it's a statement of explanation. It's what you think is the answer to your question, to your observation. Sometimes it's worded as an if-then statement. So if the temperature goes up, then the plants will grow better. That's not a good hypothesis. If the temperature goes up, the plants will grow taller, because you can measure taller, you can't measure better. If you increase oxygen, the muscle will contract 5 times faster, something like that. So an if-then statement. So you can write hypotheses that way, or you could say oxygen has a direct impact on the function of a muscle. That's an explanation of your observation. So then you have to be able to test your hypothesis. You have to design an experiment that can test for that one thing, the temperature or the level of oxygen, to what you are observing is changing. And so that's what we call our dependent variable and our independent variable, but you want to control everything else. So if you're wanting to see if temperature has an effect on plant growth, you can't give each plant different amounts of water, and you can't give each plant different amounts of sunlight or anything like that. You have to keep everything the same except for that one independent variable. So then you can say, yes, plant height, my dependent variable, was due to just that one thing I changed, nothing else. So that's how you would, parts of designing a good experiment. So then you collect your data, and then you have to ask your question. After you're done with your experiment collecting data, you organize and analyze the data, and then you have to ask the question, did we support the hypothesis or did we not? And if we didn't, we have to kind of go back to the drawing board, so to speak, and maybe formulate a new hypothesis, modify your original hypothesis, create a new experimental design with the same hypothesis. There's lots of different paths that somebody can take if their first run through did not support their hypothesis. If it did, yay, then you now have a valid explanation because you supported your hypothesis, your potential explanation for the observation, and then you can draw conclusions. And then you usually in the scientific community, you share that with people in the scientific community by publishing in a peer-reviewed journal, right? So science is usually following, science is a general word, following this format of scientific method, because if you're going to publish your results, you have to be repeatable. So somebody should be able to take everything that you did, do exactly what you did, and get similar results. And so that's the peer-reviewed part, and making the information public. Because if you didn't, if you're like, oh yeah, I have, broccoli and celery mixed together cures cancer, you can't publish that because maybe you did experiments, but if 5 other people do your experiment and they do not cure cancer, then that's not a valid result. And so we have to have these built-in check systems for science that's published. And so that's why we do the peer-reviewed journals and communicate science into this open, scientific community. Now in my subtitle here, it says scientific method ... but not always, because sometimes scientific discoveries are made outside of this kind of rigid, strict method. So I'm going to give you a few examples. So the first one is penicillin. So penicillin was one of the first antibiotics discovered, and it was discovered by accident. So Alexander Fleming in 1929, I think, he was growing bacteria. His bacteria got contaminated by a mold. And so instead of throwing the plates out and starting over, he made an observation. This observation that right around the growth of the mold, there was no bacteria. So he asked questions and he formulated hypotheses and he did more research and identified that within this mold, there is a substance called penicillin. And it is active at killing bacteria. It is successful at killing bacteria. And lo and behold, we have antibiotics, which have saved millions upon millions of lives on this planet. Another one that's not quite so awesome as penicillin, I guess it can be kind of awesome, was Teflon. Teflon was an accident. It was an industrial chemical that was being used for a completely different purpose, but the researcher who was working with it noticed that nothing really stuck to this particular substance, and so then they marketed it as a non-stick coating on cookware. And then lastly, the story behind artificial sweeteners, I don't know if Sweeten Loaf specifically for this one, I think it's aspartame, was a chemical substance that was being used for an industrial non-food item, but somehow, I don't know if the chemist was touching his fingers to his tongue, really not supposed to do that in a science lab, but somehow whatever he was working with was tasted and the individual recognized that it was a sweet taste. So they did, again, making observations for their testing, found that it was stimulated our sweet sensors on our tongue, but it was not anything that could be metabolized by our enzymes in our body. And so artificial or no calorie sweeteners is you're basically telling your tongue that it's sweet, but your body can't do anything with it. It can't burn it for energy or use it as a caloric intake. It just leaves your body unaffected. And so that is another one of these happy accidents in science. Okay. And so then the last topic that I wanted to just touch base with you on a little bit is here I pose this question with all these pictures. What do these have in common? I have the book of Albert Einstein's theory of relativity. I have the heliocentric model of the sun being in the middle of our solar system. I have a picture of some dude's anthrax-infected finger, and I have a picture of plate tectonics, tectonic plates of our planet. So what do you think? What do they have in common? So these are all theories, right? So theory of relativity, the heliocentric theory, the theory of plate tectonics, the germ theory of disease. So I wanted to just spend a minute or two on this idea of what a scientific theory is. So a lot of times in common language, the word theory is used to be like a guess, like I guess it's my theory, that's what I'm thinking, but you don't really have much information to support it. That is not the case for a scientific theory. A scientific theory is something that has been supported by multiple experiments, lots of supported hypotheses, and unless something else comes along to change this idea, this is our accepted understanding of the world. So Albert Einstein's theory of relativity, the idea that the sun is in the center of our solar system, not planet Earth, which a lot of people thought for thousands and thousands of years, that bacteria and germs actually cause disease, not a curse on your family or bad air coming from the swamp. It's actually bacteria that cause disease. And that our planet is broken up into these big plates of continent and oceanic plates that drift, and that's where we see a lot of our earthquakes and volcanoes and tectonic activity. It wasn't up until the 1960s that the theory of plate tectonics was widely accepted in the scientific community because we've finally had ways to measure and test and get evidence to support this idea. So think of scientific theories as well-supported, widely accepted explanations for natural phenomenon. Okay, all right, and the last piece here is this idea of paradigm shifts. So a paradigm shift is like some of those theories that I showed you on the last slide. So going from an Earth-centered solar system to a sun-centered solar system was a huge paradigm shift in the science of astronomy. Plate tectonics was a huge shift in the science of geology. So what are some paradigm shifts in the study of biology? Some big, like, mind-changing revelations in this class that we're going to be studying. So I have 3 pictures of old dead guys. That's usually a lot of our discoveries are done by people who are no longer with us. But the first one was Charles Darwin. We talked about that in one of our themes. His theory of evolution by natural selection just was a game changer in this understanding of how living things came to be, how they are related, how they share common ancestors, And we're getting more and more evidence as technology is increasing to support this theory of evolution. This middle guy here, we'll be seeing him a little bit later in genetics. This is Gregor Mendel. The idea that traits are passed on from one generation to the next in particular ways. So before Gregor Mendel, people didn't understand why you looked like your parents. They didn't have a mechanism for how these traits were passed on, or why you might have a 3 generations, the grandparents and the grandkids look more alike than the parents in that relationship. And so what happened? Why did that trait skip a generation and show up in the grandkids? So Gregor Mendel figured out how that happened. And then the last really big paradigm shift in biology that's listed here is Louis Pasteur, known for pasteurization, but he was also known for disproving this idea of spontaneous generation. The idea that living things can arise from non-living things was an acceptable explanation for things for thousands of, like 2,000 years, from Aristotle saying, you know, you see a dry lake bed or a dry mud puddle, rain comes down, and then all of a sudden you have worms and frogs and fish and stuff. Where did they come from? They came from the mud and the water, which are non-living. That can't happen. So Louis Pasteur did great experiments on disproving this idea that living things come from non-living things, which is called abiogenesis. That is not true. So if you're to go on to take a microbiology course, you'll learn a lot more about Louis Pasteur and the battle that was being waged when trying to prove or support or disprove spontaneous generation. All right, so that finishes the little mini lectures for chapter 1 material. Again, if you have any questions, send them via email, Blackboard course messages, or try to attend a Zoom office hour. And we'll see you next time when we get into some chemistry with chapter 2. Bye.